Hi, I'm Rebecca Duncan and I'm a PhD student with UNIS Svalbard and University Technology Sydney.
Hi, my name is Vanessa Pituzzi and I'm a PhD student with the University Centre in Svalbard and the University in Tromsø.
We've been really grateful we've been able to collaborate with the Hearts and the Ice ladies, Hilda and Sunniva. We went to visit them in March and got to see how their cabin works, how they've been living, and meet their beautiful dog, which was a really great experience and quite inspiring as well. During that time, we had a look at the ice conditions with them and showed them our equipment and taught them how to take ice cores. So we taught them how to use the ice corer and take the associated measurements like snow depth and sea ice thickness, so they were then in a position to take regular cores for us.
Since April, Hilda and Sunniva have been taking cores for us once a week. This is very important information for us as it enables us to map the biodiversity of sea ice fauna more regularly than we would otherwise be able to do and capture the seasonality. In Svalbard, this topic is highly understudied. Thus, the collaboration we have with the Hearts and the Ice ladies enables us to gather important information which is lacking, especially in the light of changing sea ice conditions. It's important information that we need in order to evaluate the effect of changing ice conditions on the Arctic coastal marine ecosystem more accurately.
Let's dive in!
I'm part of a team that is investigating both the primary producers, the ice algae, and the grazers, so the meiofauna. It's very small organisms, generally smaller than a millimeter, that live inside of the brine channels that form during the formation of sea ice. So you can imagine sea ice, the inside is basically like a labyrinth or like a Swiss cheese. It has lots of holes and channels that can be inhabited by small plants and animals. And my project is to look at the abundance, so the total number of these organisms that we find, how they fluctuate seasonally, is there difference between the sites that we go within the fjord, so if we have areas of high snow cover, low snow cover, when it's shallow, when it's deep, and also when we compare season from at the start of the season, March, to the end, May. So right now it's end of April, so this is usually when things are starting to kick off. As you can see, the sun is shining despite it being past 6 o'clock in the evening. So this is in a way the interesting season when things start really happening and exploding inside the ice.
I'm here in Van Meenfjorden to look at the ice algae. I'm interested in how changes in light penetration due to difference in ice thickness and snow depth impact the nutritional content of the ice algae. I'm interested in ice algae because they are like the grass of the ocean. They are the bottom of the marine food web, and so how their protein, lipid, and carbohydrate content changes will carry through the ecosystem, impacting the higher levels such as fish, seals, and polar bears. In fact, polar bears can derive up to 70% of their lipid content indirectly from sea ice algae sources. So whilst the algae might not be cute and cuddly, they really do matter.
At each site we collect multiple cores for both biological and physical parameters. For the biological we collect for ice algae and meiofauna, and the physical one is for salinity and temperature. After we've taken the core, we go to our sample processing station where we put the core inside of a cutting board and collect the lower 3 centimeters for the biological cores, and the whole ice core is cut into pieces for the salinity. Temperature is taken at 5 centimeter intervals, which enables us to look at the physical living conditions and space inside of the ice.
Hi, my name is Leni Runge. I'm a master's student at Copenhagen University in climate change with a focus on the polar regions. So we're trying to take a consistent number of cores, and for each core we're looking at 3 different things. So first of all, we're measuring the snow depth. After we made a hole, we just look at all the sides to get like an average. So this one, for example, is 7, 6.5, and 6.5. And then once we have made the hole with the drill, we're looking at the thickness. We're using this measure tape. And then we just stuck it in and measure the thickness of the ice. So this one, for example, is exactly 50 centimeters. And in order to measure the freeboard, we have to remove the slash from the hole, because that would give us a false measurement. So this is sometimes a very tedious exercise, but once we've taken away all the slash that just naturally enters the hole when we drill it from the snow cover, we also do that here, and that's about 1.5 centimeters.
At each sampling station, we also take a suite of water-related measurements. We start by measuring the light penetration under the ice, as this is a very important factor for both ice algae and ice fauna. Then we take water samples using a Niskin bottle to investigate the nutrient content of the water and determine if the organisms are nutrient limited or not. Then we use a CTD to take a temperature, salinity, oxygen and depth profile of the water column.
Occasionally, we will also collect zooplankton nets at some of the sampling sites to investigate secondary production in the water column during the spring, as some of the zooplankton that we find in the water utilizes the ice algae at the start of the season. As you can see, the sampling effort required to answer these important questions is extensive. It also involves us actually getting to the sampling sites, which generally means a 2 to 3 hour drive via snowmobile. Not only is this time consuming, it is also expensive and dependent on weather and snow conditions. And it increases the carbon footprint of our research. Having the hearts and the ice ladies collecting cores for us from outside the cabin, we have been able to increase our capture of the seasonality of the ice ecosystem without increasing our logistical effort and environmental footprint.
We've been really grateful to have this collaboration with the Hearts and the Ice Ladies, and it makes sense to collaborate. As Arctic explorers and adventurers, the Hearts and the Ice Ladies are passionate about understanding what's happening in the Arctic and how climate change is affecting it. And as scientists, that's also what we're passionate about, exploring and understanding. So together, we're using our independent skill sets to not only do the science and do the research, but then to spread these messages to the public. So it's been a really fruitful collaboration, and we're looking forward to hopefully some more work together in the future.